The osmosis required practical will probably come up on your biology exam on Tuesday, so let's bag some marks. Now, the purpose of the practical is to determine the unknown concentration of some sort of plant. It may be a potato, a carrot, beetroot. I've seen all of these come up in an exam before. And to do this, we use our knowledge of osmosis, which is the movement of water from a high to a low water concentration across a partially permeable membrane. Now, imagine our substance, our potato, is the Goldilocks of the situation. We need to give Goldilocks a range of different concentrations to choose which is hers. That is our independent variable. The thing we change is the concentration of the solution. So, we take our substance, our potato, we cut many chips, all of which are the same size and from the same potato. We then place them onto a balance to find their starting mass, and then put each of the chips into the different concentration solutions to let osmosis go to work. Now this brings me to my dependent variable, which is the thing we're looking to affect, and in this case that is the percentage change in mass after we take these potatoes back out of their solutions and reweigh them. All that's left to do now is to analyse our results. We need to plot a graph of the different concentrations and the percentage change in mass. You'll notice when you plot the points, there will be an intercept with that concentration axis. And it's that intercept that we can identify as the unknown concentration we were looking for of our substance. Now think about the science behind why that is. At the intercept, there is no gain or loss of mass. That means water has not moved in or moved out. We know osmosis means that the water moves from where there's lots of it to where there's not so much. So if it hasn't done that, it means just like Goldilocks, we have found the sweet spot. We have found where the concentration inside and outside of our substance is exactly equal. Now often in exams they like to ask you one last thing about the control variables, things you need to keep the same in your experiment to ensure that your results are reliable and valid. A few things you could say the volumes of the different concentration solutions that we put those different substances into. The substance itself, you want it to be from the same species, have the same shape and size, each of those chips. You also want to keep them into the solutions for the same amount of time.